All right. Magandang umaga po sa lahat. Kuya Jeff, can you share the Canva? Okay, so I think we're now live on Zoom. Magandang umaga po sa mga participants natin dito sa Zoom and on YouTube. We're now live on YouTube. And yep, welcome po sa special topic number 10, if I'm right, of the FilSciHub Research University, and it's titled Computational Chemistry and its Application. So ito po ay bunga po ng clamor dito sa subject po na ito, sa topic na ito. And this is following sort of like a series of data analytics series of the FilSciHub Research University, and we are very proud to have the group from the Good Vibes Laboratory, headed by Professor Nellas, to join us today and discuss about this computational chemistry and some of its applications. So, next slide, Kuya Jeff. So, before anything else, we would like to remind everybody to kindly mute. Pag minsan po nadidistract po yung speaker natin when we suddenly barge in, if we are not muted, and kindly turn off your video for now para po hindi po lumaki yung bandwidth natin. And you can turn on your video later if you want to post a question or a comment, and we will recognize you and we will let you speak. Okay, so no recording of the video, please, because the entire recording of the webinar will be posted on our website right after the webinar. Okay. Okay. Good morning po. Certificates of participation is always issued for all of our webinars, and we will post the Google form link on the chat box here in Zoom, and as well as in YouTube, and also in the video description in YouTube pagkatapos po ng webinar. So, dun po sa mga nagtatanong, lahat po ng webinars namin ay may certificate of participation, and you can always just check the video description in YouTube pagkatapos po ng webinar. Okay? So, in order to get or obtain your certificates of participation, you must be a follower of our FilSciHub official YouTube channel, and also our Facebook page. Okay. So, to introduce our group, dun po sa mga bago natin na participants, I would like to usher in our founder and CEO, Dr. Jeffrey Bonkin, who is joining us from Houston, USA.
Right.
Doc Jeff.
Can you hear me?
Yep.
All right. So, ako po si Jeffrey Bonkin. Ako ang founder ng Filipinos. Ako po ay nakabase dito sa Houston, Texas. So, magandang umaga po sa inyo, magandang gabi dito. So, Filipino Science Hub. So, this is an online platform that I founded back in 2012, and its main objective is to promote the cultures of STEM education and research in the Philippines. So, ang target audience po natin dito ay mga estudyante at mga guro sa high school at sa kolehiyo. So, meron po kaming isang big goal, and this goal is to actually launch or to help build a strong culture of innovation in the country. And how do you launch a culture? So, meron po kami dyang dalawang parallel approaches. So, the first of which is to strengthen the foundations of STEM literacy in the country. So, kailangan yung mastery natin ng ating iba't ibang STEM topics, kailangan mas mapaangat pa natin para makasabay tayo sa ibang mga mas mauunlad na bansa. But then, for us to be able to launch a culture, what we know should be actualized. So, kailangan na-apply natin to create value for the country. So, dito na po papasok naman yung aming program, focusing on research education. So paano ba yung mga fundamental steps in research? How do you actually apply the things that you learn inside the classroom to innovate? So ito po yung dalawang main programs namin sa Filipino Science Hub, and this is actually covered by two main programs. So the first of which is Filipino Science Hub Education, and then the second one is Filipino Science Hub Research University. So ito po yung mga idedetalye namin in the succeeding slides. But then before we actually go there, for those of you who may be wondering, okay, what do we do? So I believe that, you know, like our contents and the value that we create are actually more best represented by the web events that we conduct. So over the past 12 months already, we have conducted about, the numbers here are not necessarily, not really updated. I apologize for that. So we have conducted about 30 web events already. And those events are comprised of teaching webinars. So meron po kami mga training courses for teachers on how to teach, you know, in the new normal mode of education. So meron din po kaming career guidance talks for both teachers and students, web lectures and tutorials. So again, as we said, we'd like to elevate the mastery of STEM courses. So meron po kami mga Tagalog English lectures in different areas of the sciences. We also conduct a research training courses. So dyan na po yung research webinars namin. So JP will talk about that later. And then lastly, STEM special topics. So obviously I have to update this because this is actually the 10th special topics webinar that we're delivering since we started. So ito po yung mga ginagawa namin ngayon. The good thing about these webinars is that they're all uploaded on our on our YouTube channel. So dun sa mga topics po that you may deem relevant to your interests, you can always go to our YouTube channel and you can just like watch them. Wala naman pong bayad. Lahat po ng aming web events and training courses ay wala pong bayad. Okay. So I would like to talk about FilSciHub Education. So this is the first program that I mentioned earlier. So most of our projects right now are focused on empowering our teachers. And why is that? So in order for us to be able to reach as many students as possible and to influence them to pursue a career track in the STEM area, in different STEM areas, it's very important that we equip our teachers because they are the ones who can, you know, spread out, you know, the word. So at the moment we have about nearly 32,000 followers on Facebook. And majority of them are actually early to mid-career level teachers, so educators. So if you can imagine that, about 30,000 teachers are to be trained by organizations like us, you can just like imagine the impact of that. So for example, if each of those teachers would teach for 30 years on average, and if they could influence 30 students per year, you're looking at about 27 million students impacted over a span of 30 years. And if only 1% of that would decide to become a scientist, you're looking at about 270,000 Filipino scientists or researchers to be that can be produced like over the next 30 years. So that's actually more than what we need at the minimum for us to be able to keep up with our neighboring countries in terms of innovation. So ito po yung talagang tinatrabaho namin sa Filipino Science Hub, and it's a team of different Filipinos. It's a team of Filipino scientists who are actually voluntarily working together para po matulungan ng ating mga guro at ang ating mga mag-aaral. Kailangan pong mas dumami yung mga kaparehas namin at kaparehas po na ating mga speakers ngayon. Okay, so what do we do in FilSciHub Education? So as I have mentioned earlier, we are conducting teacher trainings po. From April up to September of last year, we have conducted a series of teaching webinars that focus on how we can actually further motivate our teachers. We've been we've been sharing with them like tools that they can use to transition to the new normal mode of teaching. And we are also providing them with guidance in terms of how they can enhance their ways of teaching in such a way that the lessons are delivered in the most impactful manner. So ito po, we've been inviting educators from different parts of the world, Filipino educators from different parts of the world, to share their best practices po. So ito po ang aming binibigay, and we have a follow-up program on that, which I will talk about in one of the succeeding slides. Okay, so but very recently, over the past 3 months, this is most relevant to our high school sector. So we have recently launched FilSciHub Education's modules program. So essentially what we do here is on a daily basis, we are rolling out teaching modules in the areas of biology, chemistry, and physics. So ito po, these are carefully created and curated by graduates of Bachelor of Science in Mathematics and Science Teaching from the University of the Philippines. So each of the module that we actually roll out contains teaching guides, prescribed classroom activities, as well as problem sets. So para po talaga meron tayong tools for assessment. So these are things that we roll out on a daily basis. Over the past couple of weeks, naka-break lang po kami. We're re-strategizing to further improve our contents. But check out our website and our Facebook page kasi lahat po ng mga nauna naming modules ay naka-upload naman po, and you can access them and download them for free. Okay, so this is...
The Filipino Science Hub is calling for applications for the pilot run of the STEM Teaching Fellowship Program. This program is devoted to early career educators to learn innovative to generate educational materials that will be shared on a national platform.
Okay. JP, may audio pala yun, na-surprise ako. So by the way, meron po kaming bagong program on teacher training. So this is a formal teacher training school that we are launching. Magpa-pilot run po kami this year. And this is actually a collaboration among 3 different organizations. So it's Filipino Science Hub. It's the foundation for the upgrading of the standards of education. So that's the foundation of the Lusiotan group of companies po. And then we're also collaborating with the Philippine Normal University. So ito po, this is a fairly rigorous training program for high school teachers po in math, chem, bio, and physics areas. So we'll run courses over the next 4 months, and then after that, meron po itong mechanism for internship para sa mga teachers. So we actually have recently posted a call for application. So check out our website and our Facebook page if this is something that may be of interest to you. But in particular right now, the pilot run will focus on high school educators, and hopefully, those who are within the early year career experience level, so 0 to 5 years po of teaching experience. So mali, kung interesado po kayo dito, and we will formally launch this like one of these days po. We're just finalizing more of the details, with PNU and with Fuse, but this is really something that I highly encourage our teachers to participate in. All right. So that's essentially Filipino Science Education. Now I will turn the floor over to JP for him to talk about FilSciHub Research University.
All right. Thank you, Kuya Jeff. Good morning po sa lahat. I think we are now full here on Zoom, and I'm sorry po dun sa mga hindi na nakapasok, but we are now live from YouTube, so you can always participate in YouTube, and you will get the certificate just as well. Okay, so segueing to FilSciHub Research University. So ito po yung current program po namin to promote the research culture among our students and teachers. And this is actually a webinar series focusing on research, on scientific research, and all of its different aspects. Okay, and here we will share our own research experiences and some best practices when carrying out a research project. And we'll also share some tips and advices on that. Next slide. So FilSciHub Research University is composed of 8 mandatory courses. So inisa-isa po namin yung lahat ng we itemize the steps for the different research projects. Like step 1 is the research ideation, literature review, experimental design, all the way up to poster and conference presentation. So this is very some sort of chronological order, but yeah, we are now on to the sixth topic for Research University. These are the mandatory courses. So next week we will have the topic on scientific communication, or uh, how to write a scientific report, which will be headed by our founder and CEO, Doc Jeffrey Bunkin. So uh, if you complete all of these courses, so you can participate in past courses and still get a participation certificate for that. And if you complete all the mandatory courses, we will issue a completion certificate. Okay, next slide, Kuya Jeff. And yung mechanism po, the mechanism for issuing the completion certificate is summarized in this info webinar last January. And doon po, we we gave a step-by-step guide on how to join the Google Classroom, because that's the mechanism by which we issue the completion certificate for Research University. And we will also send the link to the Google Classroom so that you can participate in this program. Okay. Next slide. So FilSciHub is always present in all of our platforms, mainly our website, www.filscihub.com. And we're also on Facebook and YouTube and even on TikTok. And well, mainly our platform is our website, which contains all of our webinars, tutorials, modules, virtual lab, and features. So if you miss anything, you can just check our website at www.filscihub.com and just browse through all of the sections there. And ayun po, you can always catch up on anything that you might have missed. All right. So today, we have another special topic sa FilSciHub Research University. So this is computational chemistry and its applications. And today, we are joined by the group from UP Diliman, the Good Vibes Laboratory, headed by Professor Ricky Nellas. And I would just like to introduce our speakers for today. Professor Ricky Nellas, or Dr. Ricky Nellas, is a UP scientist from the Institute of Chemistry at University of the Philippines, Diliman. He is the Associate Dean for Research, Innovation, and Development, Enterprise and of the College of Science at UPD. He's also the principal investigator of the Virtual Biochemical Explorations Laboratory, or Good Vibes. And he has authored over 40 academic publications here in the Philippines and also abroad. He finished his PhD in chemistry at the Louisiana State University in USA. And he is a graduate of Bachelor of Chemistry from the University of the Philippines, Los Baños. Okay. Our next speaker is Almira Ocampo, who is a researcher and an MS chemistry student at the University of the Philippines, Diliman Institute of Chemistry. And she's one of the principal researchers at the Good Vibes Laboratory. And she has been an academic tutor at the St. Calungsod Tutorial Center. And her affiliations include the UP Association of Chemistry Majors and Enthusiasts and the UP DOST Scholars Association, in which she's very active in. She finished her Bachelor of Chemistry at the University of the Philippines, Diliman, and at the Casimiro Inares Senior Memorial National High School. For her high school degree. We also have Jokent Gaza, who is also a researcher and MS chemistry student at the Good Vibes Research Group. And he specializes in dynamics of biochemical systems and force field development. And he graduated from the University of the Philippines with a bachelor's degree in chemistry, cum laude. And from the Subic National High School Special Science Curriculum with first honorable mention honor. Okay. So, maraming salamat po sa ating mga speakers. And if you can go back to the... Yeah. So, maraming salamat po sa mga speakers natin and all of you joining us today. And I think at this point, Professor Ricky can already share his slide.
Good morning. Set up po na.
Good morning po. Okay po. So magandang umaga po sa ating mga YouTube participants from Pangasinan, Tarlac, La Union. I'm just reading the stream here.
Yeah, we actually have about 300 participants today.
Yep. From Binyan, Romblon, and we also have some international participants. So I think we should stick to English at this point. North Cotabato. Ayan, from Bulacan, si Miss Alma Abra. So dun po sa mga nagtatanong about membership. So we don't have a particular membership for FilSciHub. And but you can always sign in or register for our programs and campaigns. So right now we have FilSciHub Research University, and soon we will have the well, I don't want to preempt it, but yeah, we have an offering for our teachers o dun sa fellowship namin.
So if you're asking about membership so we're building two separate communities within our main program. So yung FilSciHub Research University, we currently have 1,000 students in that program. And once you finish all those courses that JP talked about earlier, you'll be given a completion certificate. And what we're trying to build there is a community of FilSciHub followers who have been formally trained in our research program, and then after that we'll take care of that community. So we plan to conduct specific webinar topics that will cater to their various research interests. So that's a research community that we are building, and then we're also doing the same for our education sector. So yun pong training program na sinabi namin sa inyo, we're gonna start with the pilot run, but then we're scaling it up on a nationwide scale. So we're really also trying to build a network of teachers who can work together in generating high quality teaching materials para po lahat tayo sa Pilipinas magagandang ginagamit. Ayun.
I think Sir Ricky is ready with his slide.
Okay.
So Sir Ricky, take it away.
Yeah. Can you hear me?
Yes po, sir.
Okay. Okay na rin yung slides. Okay. Good morning sa lahat. Thank you for participating. And to Jeff and the group, thank you for this platform. And thank you for inviting our lab, our group. Okay. So, to start with, ito yung group namin. Yeah. Masaya at may kakainan. Okay. And hopefully, we can introduce computational chemistry in the fields of chemistry. So madaming computational science, meron computational physics, meron din computational na data analytics. So yung focus lang natin ngayon ay computational chemistry, at in-invite ko yung dalawa kong RA master students para at least makita ninyo kung anong ginagawa ng bawat isa. And then we will share the entire endeavor ng buong Good Vibes Lab. So first, I'll start with introducing the lab as well as the researchers, and then focused lecture with Jokent and then with Almira on different topics, and then i-close ko with endeavors ng lab and then future directions ng research namin. Hopefully, magkaka-interest kayo, and then Kung papasok kayo sa UP Diliman, then pwede namin kayong maging part ng lab, or kung hindi man, we can always be collaborators. Okay, so computational chemistry. So chemistry pa rin to. Okay, this is still chemistry. It's in the different dimension lab. So we always hear about chemistry in the lab, the experimental part. So this time we will hop into another dimension, which is the computational chemistry. Sometimes we call this in silico chemistry. In silico meaning in computers, so in hardware. So we use computers here to do experiments, mimic experiments in a real chemistry world. So basically, the computational chemistry is the computer parts, and then we use modeling, we use predictive measures using computers so that we can guide we can guide our experimental people, or they can also give us feedback on the limitations of our science, and then it can go back and forth to improve our set of knowledge as well as skills. So the applications for computational chemistry involves computer-aided drug design, mechanistic insight on chemical reactions. We also have protein design, prediction of structures of proteins, and then modeling of reactions, as well as how to do synthesis in a very nice way. Again, experiment to computer, computer to experiment, feedback lab. At least magkaka-collaborate yung experimental wet and dry part ng chemistry, and then pwede natin ma-improve kung ano man ang kaya i-improve. Limitations for this, may limitation ng computational chemistry, so hindi lahat kayang gawin ng computational chemistry. Hindi rin lahat ng ilalabas ng computational chemistry ang magkakatotoo sa experiment. So basically, we should look at the insights of what computational chemistry could do. Okay, limitations could be complex in vivo systems. Pag sobrang komplikado na yung system, hindi na kaya ng computational chemistry. Or kailangan mag-develop ng systems to handle this one. Okay, some computational methods rely mostly on empirical data. So kung wala kang data, wala ka ring mapaprocess. Non-linear relationship between computing power and protein size. So nagdidepende tayo sa kapangyarihan or power ng ating computers to crunch numbers. Pag sobrang hirap na talaga, sobrang laki ng system na tinitingnan natin, hindi na kaya ng computer. So yan, limitation din yan. And how to correlate our theoretical results or computational results with experimental results. Okay? So anong gamit ng computational chemistry sa modern society right now? Kung titingnan ninyo, merong tinatawag na computational or in silico vaccinology. Meron ding mga chemicals ngayon or mga drugs na nasa pipeline to combat SARS-CoV-2 or the COVID-19. Most of these works are computational bago pumunta ng experimental. May bagong results ngayon na even with amino acid sequence, kaya nang i-predict ang 3D structure ng ating mga protein. And this is very important kasi yung mga sakit natin ay usually to be played using different machines or the protein machines. So kung naintindihan mo yon, pwede mong paglaruan, pwede mong i-intervene. And also there are a lot of innovations or discovery of materials na yung simula ay computational. So dati, trial and error lang tayo. Ngayon, medyo magastos yung trial and error. That's why we sort of use computational chemistry to guide experimental aspects. And then also for our drug discovery, nasa pipeline ng in silico or computational para mapabilis at maayos yung ating drug discovery processes. Ano magiging trabaho natin pag nag-computational chemistry tayo? So basically, dalawa. Pwede kang pumunta sa academe or the education or the industry. Pag nasa academe ka, pwede kang maging ako, principal investigator, which you do this stuff. Develop models of biochemical systems, innovate current computational models, collaborate with colleagues from around the world. Pero kung bata ka pa at kagagraduate mo lang ng BS or MS, then you can be my research associate. I will also give you the chance to do some works of the principal investigator para ma-prepare ka for future work being a principal investigator or the head of the lab. Sa industry naman, yung sinabi ko doon, pwede ka maging laboratory chemist din na computational. You perform this stuff. Or kung gusto mo, doon ka sa software development. paano mapabilis yung processes, paano mapaganda, paano gumamit ng machine learning, paano gagamit ng artificial intelligence. Okay, so dito sa industry, meron din tayong pwedeng pagtrabahoan. And these are the necessary skills. Pwede magsimula sa zero. Again, huwag matakot. Pwede ring matuto ng mabagal. Pwede ring matuto ng mabilisan. The only thing is you should learn along the process and then try to do your best. Necessary skills is knowledge of basic programming languages. Pwede itong matutunan along the way. Nagsimula din akong zero. Wala akong alam dito. And then I have to force myself to learn kasi kailangan for data processing. And then you should be aware of strengths and weaknesses ng mga computational software para alam mo kung anong software ang gagamitin. At kung alam mo na yun lang yung available software and may limitation siya, pwede mong i-improve. And lastly, must be able to interpret the results of calculation and simulations. Dito papasok yung ating high school chemistry, high school science, tapos yung basic natin, foundational chemistry sa BS courses natin. Okay. So, wala kaming chemicals na ginagamit sa lab, pero kailangan namin to. Kailangan natin ng computers. Kung may laptop kayo, may computers kayo, hindi ko alam kung anong capability ng laptop or computers niyo. You can have one brain or until 4 brains in one of your computer. And that's not enough to process computational experiments. We have 1,000 to a lot of computers working as one. So ito yung tinatawag nating high performance computing clusters. So meron tayo sa UP Diliman, meron kaming sarili sa lab, at meron din tayo sa Pilipinas. Ito yung tinatawag na DOST-ASTI, na sangkatutak na computers ang pwede mong gamitin, sangkatutak na computational power ang pwede mong magamit when you do our computational experiments. So kung mabagal to, mabagal din ang experiment mo. Kung luma na yung computer mo, baka abutin ka ng siyam-siyam. So mahirap talaga kung wala to. And I think one of the priority agenda to ng DOST, since DOST wants machine learning as well as artificial intelligence. So processes naman, ganito lang yung process sa computational. I will be talking about 2 things lang dito. Sorry. Yung docking lang at saka molecular dynamics. Okay. Yung training ko talaga sa postdoc is biodynamics. Medyo sa higher level na yon. Later, madi-discuss naman ni Almira at saka ni Jokent. But basically, kung wala kang protein sa database, wala kang crystal structure at amino acid sequence lang yung meron ka, so hahanapan mo siya ng 3D structure by the use of homology modeling. There are softwares where you can do this ab initio. And then pagagalawin mo yung protein para makikita mo through time kung anong pagbabago ng mga structures ng proteins na yun. So you can do molecular dynamics. If you have a lot of structure for this one, and you have a ligand, or this is what your receptor is, then you have different ligands, or what we call as drug candidates, then you can do ensemble docking. And kung hindi masyadong maganda yung dikit ng mga compounds mo, then babaguhin mo siya. And paano mo siya babaguhin? Dapat babaguhin mo siya na hindi trial and error din. So you have some sort of a modeling, which is we call as 3D quasar, to do that. And then you can do modification of your compounds or your leads. So ito yung process para makakita ka ng magandang match between receptor and your ligand. And dito sa modification na to, dito sa analysis na to, kaya din i-determine kung yung makukuha mong compounds or drugs ay medyo toxic or not. So you can do admitox studies, and also you can also tell the experimentalists na medyo mahirap or madaling i-synthesize yung compound na yun. Therefore, giving insight on how we should proceed with the drug discovery. So gagamit din kami ng molecular dynamics. Kung titingnan mo yung gumagalaw dito, pwede kang magkaroon ng initial structure at in different conditions or through time, pwedeng magbabago yung structure na yan. Ito yung tinatawag nating biological machines. Lahat ng nangyayari sa katawan natin ay governed by biological machines. Lahat ng sakit pwedeng i-intervene if you know the biological machine that you need to intervene. So medyo komplikado to. At ito yung reason bakit hindi, kahit may in silico na, hindi pa rin sobrang mabilis yung drug discovery natin. Okay, so kailangan ng magandang matching dito, magandang monitoring ng pagbabago ng protein natin, and then hopefully, okay, kaya nating i-decipher kung anong pagbabago ng mga structures, and pag nagbabago yon, ano ang nangyayari sa mga drug candidates natin or ligand. So this is my lab, or this is our lab. We call it Good Vibes. Vibes for virtual biochemical explorations. It's good because we don't do anything bad. So that's good vibes. Okay, I did my BS in UPLB. Okay, through a scholarship. Kung wala yon, wala rin ako dito. PhD in LSU through assistantship. So in Baton Rouge. And then to further my work. So I work on nucleation here and then biological systems on postdoc with a joint postdoc from the University of Tennessee, Knoxville at the Oak Ridge National Laboratory. So ito yung interest ko ngayon. Naging interest ko dahil trabaho ko nung PhD ako at saka trabaho din nung postdoc. So we have multi-component atmospheric nucleation. So tinitingnan natin paano mag-aggregate at paano yung hangin nagiging ulan at anong kasama ng ulan na yon. Pag istay bumagsak at doon may intindihan natin yung mga acid rain concept as well as impurities in your rain water. Okay, so you can also have aggregation of biomolecular and supramolecular systems para naman to sa mga biomedical approach applications. Then you have complex salvations, bioenergy, and mahirap makipag-deal with experiments kung hindi mo alam kung paano interaction ng bawat molecules. So kailangan kong mag-design ng force field mag-design ng force field. Si Jokent ang aking tao dyan for force field development, and he worked with Taiwan with this. Tapos, sa clamor naman ng mga experimentalists kasi ang laki ng drug discovery ng UP Diliman or ng DOST with the entire Philippine researchers. So, tumulong na rin kami sa design ng mga molecules. So, we also started our computer-aided drug design. So, we work mostly on schistosomiasis, finding drug for this, small molecule drug discovery, development of force field, and then playing with biopesticides. Using DA Biotechna funds. And then we also do protein engineering to harvest some sort of important chemicals. So mag-talk ngayon si Jokent. Jokent is a master's student in my group. And then he is into doing force field development. Medyo mahirap to. So he worked with Professor Chai at Taiwan. And then he does a lot of conotoxin studies on biomolecular simulations to determine different structures of peptides, conotoxins in different system conditions. And then ito yung protein-protein docking. So pag pumasok yung SARS-CoV-2 doon sa katawan natin at pupunta sa ACE2, basically what you are looking is a protein-protein interaction. So alam din ni Jokent yan. So bibigay siya ng konting talk mamaya-maya after this one. And si Almira naman is our biopesticide expert. So nagdi-design siya ng compounds or ng small molecules to have a better interaction with a receptor from different insects. And for this one, yung ipapakita lang niya yung sa rice weevil. Okay? So may mga publications na si Almira at saka si Jokent, so you can listen to their talk or the part of this talk, and then you can also take a look at the publication kasi nandoon yung details. And my group is open for collaborations, and then you can always email us for concerns. Okay, so let's have Jokent now.
Yep.
Good morning po. Good morning. Narinig po ba ako?
Yes.
Okay. So, good morning
Take it
all
away.
po. My name is Dr. Ken Gaza, and I'm here po to present our work entitled Visual Screening of potential anti-hypertension drugs and in silico study on benzimidazole compounds, peptide ligands, and natural products. But before that po, I would like to first formally introduce myself and give some talk regarding my experiences while doing computational chemistry. So I'm an MS chemistry student po currently studying at the University of the Philippines, Diliman. And for my undergraduate course po, I took BS chemistry in the same university. And I'm an aspiring computational chemist who wants to specialize on force field development and the dynamics of biochemical systems. So I've actually been doing computational chemistry related studies since the first semester of my third year as a BS chemistry student around 2015, so 16 years, 6 years now. So for why I chose this field, it is because all of my interests as an aspiring scientist seem to gravitate towards computational chemistry. So I've always liked chemistry, math, physics, and of course, like computers. I actually consider myself as an epitome of 2000s, early 2000 kids that grew up with their eyes glued in front of their computer screens. So, and I also consider myself fortunate enough to study biochemistry under Dr. Isidore Yu, one of our lab's collaborators. So, one of my favorite biochemistry courses was about proteins and the relationship of protein structure with its function. And luckily, again, by the end of that course, I've decided na I want to pursue this topic, and I decided to join yung Love of Dr. Ricky Nellas, which actually opened on that same year. So, yeah, so luck din yung naging experience. So for my first project, which is also my undergraduate thesis, it is about anti-freeze protein. So what we did here was we used computational method called molecular dynamics to study the behavior of ice in the presence of an anti-freeze protein. And we mainly focus on the structure of the ice form, the structure of the protein, the and the thermodynamics of the ice formation process. So the one on the left is the ice, the one on the center is the protein, then yung sa right is yung water in liquid form. And we saw na due to the presence of the protein in the center, na-stop yung formation ng ice to the right side of the box. So for my MS thesis naman, I'm currently working on the force field development of copper cofactors. So for the jargons, force field refers to the energy function and empirical parameters that dictate the results of your computer simulations. Like think of it as the DNA of your simulations. So and copper cofactors naman refer to copper ions present in some proteins. So what we want to do here is to provide the parameters or the necessary numbers that would allow us to accurately study copper proteins using atomistic methods. Other than my thesis, I've also worked on different projects under the supervision of Dr. Kenelias. Two of these projects have been published in international journals, and both of which are about neurotoxins. Neurotoxins refer to neurotoxins present in the venom of cone snails. Cone snails are prevalent in our marine environment, and our hypothesis is that given the neurotoxic activity of neurotoxins, they can serve as templates for new therapeutic drugs against different neurological diseases. And both of these articles involve prediction of protein structure at different conditions using mainly molecular dynamics. That's it for my intro. Next slide. Sir. So I would like to talk about something else naman po. I would like to talk about hypertension or high blood as we commonly know it. So in a study done by WHO, they showed that the leading causes of death in the Philippines are diseases related to the heart or cardiovascular diseases. So about previous slides po, sorry. So about for all the tabulated deaths from 1994 to 2018, about 35% of which are due to cardiovascular diseases. And another alarming statistic is the first bullet po. So about 1 in every 6 Filipinos or 1 in every 3 Filipino Americans suffer cardiovascular diseases. So if we have 300 participants in this webinar po, we can expect about 50 of us have hypertension, but of course, pwedeng mas mataas, pwedeng mas mababa because that is the statistics for the general population. And this high number has been linked to bad health habits such as alcohol consumption, smoking, and poor diet adaptation. So In another study naman po done by Dr. Sison, a survey done by the Philippine Heart Association showed that hypertension is the most prevailing CVD, followed by stroke, then coronary artery disease, and heart failure. About 38.6% of CVD is hypertension. And through complete coincidence po, next slide, sir. And through complete coincidence, so May is the Hypertension Awareness Month here in the Philippines. So I would like to remind everyone to do the following or to reduce the risk of hypertension. Remind din po yung family members, relatives, and friends that to reduce risk of hypertension, you can reduce salt intake, engage in regular physical activity, maintain a healthy weight, eat fresh, natural fruits, don't smoke, and follow medical advice. In the same study of Dr. Sison po, they released data, the latest data about hypertension, and for the people that took the survey, about 6% is a treatment rate, compliance is 57%, and the BP control rate is 20%. So yeah, again, remind po everyone to always check your blood pressure since no symptoms doesn't mean regular blood pressure. For people naman po that cannot prevent hypertension or suffer na from this disease, meron tayong different medications. In the Philippines, the main medications po are beta blockers, CCB, angiotensin receptor blockers, ACE inhibitors, and centrally acting agents. So I have these two lines po underlined because, next slide po sir. In our study po funded by DOST PCHRD, we aim to design novel drugs with similar activity as those underlying medications. Again po, the title of this project is Virtual Screening of Potential Antihypertensive Drugs, an in silico study on benzimidazole compounds, peptide ligands, and natural products. So this is a collaborative work po between our laboratory and Dr. Evelyn French's laboratory in Mindanao State University, Ligan Institute of Technology. And this work has two main objectives. One is to provide a list of potential antihypertensive drugs from a set of benzimidazole compounds, short peptide ligands, and natural products. And to design an antihypertensive drug, antihypertensive benzimidazole-based pharmacophore. So pharmacophore refers to a template structure that have significant activity towards different proteins of interest. So remember po, I have two lines underlined in the previous slide. So for this study, we have two protein targets, the human angiotensin converting enzyme or ACE and the human angiotensin receptor. So next slide please. So in this study, we have our methodology for this study can be divided into four main parts: structure preparation, molecular dynamics, molecular docking, and video freezer functionalization. So for structure preparation, next slide. So for structure preparation, we want to clean or modify our proteins of interest for the succeeding parts. And if you want to do this kind of study po, my advice po for this step is to read a lot about your proteins po, because proteins tend to have key features that dictate their structure and activity. And you have to make sure na these features are preserved in your simulation and docking studies. So for example po, this human ACE structure po was obtained from the protein data bank under the ID 108A, and its key features are: one, it's a metalloprotein with an active site that contains a zinc ion, and two, some of its residues are glycosylated. And for human angiotensin receptor naman po, the protein is a membrane protein. So in our simulations po, we have to embed it inside by layer of phospholipids. And there are different ways to prepare your protein. And if you're interested, you can visit for this site. This is a web server po, this Charm GUI web server that outputs input files for different computational methods. And I think kailangan nyo lang po ng account to use this web server. So next slide po sir. And our next method naman po is MD or molecular dynamics. So has anyone po here watched like a physics simulation video in YouTube? Like for example, how like Minecraft blocks will explode in a realistic manner, and or how Tetris or how soft Tetris blocks stack on each other. So like this is exactly like that, but with proteins and atoms. So basically, we allow the simulation of movement of atoms in our proteins, and after some time, we analyze the resulting protein structure as dictated by the position of the atoms. And if you're interested po, you can search molecular dynamics in YouTube to see kung paano gumagalaw yung atoms natin. And if we commonly do this step to obtain a good statistical representation of our protein structure. And again po, advice po, ito po yung pinakamatagal na part. So for 100 nanosecond, 1 nanosecond is, 1 nanosecond is 10 raised to negative 9 seconds. But we need about 1 month to simulate 100 nanosecond all atom MD of this protein size. So this is yung matagal na part. So make sure po na before you perform MD, tama lahat ng parameters natin. And this figure po presents yung original structure for subfigure A, and then subfigure B refers naman po to the top 10 most populated cluster structures po superimposed to each other. And next slide po, you can actually see po how the atoms move in this figure. So the one on the left, the structure on the left is the original structure, the subfigure A, and then subfigure B naman po is the structure, the most probable structure after the simulation. And then when we superimpose po those two conformations, we saw na some positions or some atoms have different positions. Specifically po, yung glycine ay glutamic acid po, T84, and we saw na mas malapit siya in the zinc ion for subfigure B compared to subfigure A. So we can correlate then this difference in protein structure to protein function and the results of our docking calculations. Next slide please. So for next method naman, we performed ensemble molecular dynamics, molecular docking of 3 different ligand types. So we have short peptide ligands with and without PTMs. PTMs are post-translational modifications of different amino acids. And then African and Philippine natural products, and also synthetic benzimidazole compounds. So yung benzimidazole compounds po are compounds po synthesized by our collaborator po in MSc. So for the jargons ng molecular docking. So in high school po we learned that molecules have what we call intermolecular forces of attraction, or IMFAs, and some atoms are attracted to some atoms but also repelled by others. So basically molecular docking po, here we try to determine this type of interaction and we try to quantify it to different docking algorithms. So for this figure po, the blue ribbons represent the proteins, the purple sticks represent the residues in the active site, and the orange stick naman po represents the potential anti-hypertensive docking. And in molecular docking, we determine the best possible docking pose and the best docking binding energy docking result. Next step. So for the last step, we perform 3D QSAR and functional fission studies. So 3D QSAR or 3D quantitative structure activity research is basically looking at your DAC ligands and then determining common structure or parts that can be correlated to good binding activity or ligand activity. So for example, yung figure on the left suggests that more steric groups, more like hydrophobic groups can be added in this portion of the ligand. While for functionalization naman po, we designed a pharmacophore for benzimidazole anti-hypertensive drugs based on our docking and TDQC results. And we concluded that we can add polar and aromatic groups at these two sites based on our late docking results. Next slide, please. And actually, I've discussed some of our results during the methodology section. So here I will just like to present some quantitative results of our docking studies. So for our peptides, we saw that aromatic groups or aromatic amino acids tend to have better affinity towards protein targets or combinations of uh, uh, aromatic groups. So, as you can see po na our, our data or our binding energy data are in negative. Negative po since that implies na meron tayong attractive forces between our ligand, our peptide ligands and our proteins. And next slide please. And we also saw these features in natural products. Natural products that are bulkier and more aromatic tend to have better binding affinity towards both ECE and the angiotensin receptor. Next slide please. And then this is a result for the synthetic compounds. So we saw na there's a template for this 13X template that has good 13 and 23 templates that have good affinity po towards towards our ACE and angiotensin receptor. And we saw na aromatic din siya and mainly free na. So what we want to do here then is to further enhance yung binding relationship po between our benzimidazole compounds and ACE and angiotensin receptor. And that's it po. I would like to thank UP and Good Vibes Live for allowing us to study this project. And then this project was funded by the OST PCHRD DBHP program. And for our computational sources naman po, we commonly use our servers located in the UPCSRC and also the HPC facility provided po by the OSTST core. That's it for my presentation.
Okay. Thank you, Jokent. So, dun po sa mga participants natin, kindly reserve your questions at the end of the presentations. And ayun, magkakaroon po tayo ng discussion later. Okay? So I think Almira is ready. So, kindly pagpasensya nyo na lang po kung hindi po namin mapapakita yung video ng mga speakers kasi nagkakaroon po tayo ng connectivity problems. So, ayan. Almira, are you ready?
Okay po. Narinig niyo po
Ayan,
ba?
yan. Okay. We see you.
Okay.
All right.
Okay po. Okay, so good afternoon, morning everyone. So for this part po ng webinar, I will be talking about an example on how computational chemistry can be used in solving problems in agriculture. Specifically, I will be talking about a research study po on how monoterpenes can be explored computationally as a potential biopesticide for rice weevils. Okay, next slide, sir. Okay, so na-introduce na po ako, but I would like to formally introduce myself. So I am Almira Ocampo. I'm currently taking up my master's in chemistry in UP Diliman, and I graduated last year with a BS chemistry degree, where I took my first year at UST and finished my degree in UP Diliman. During my undergraduate years, I was an RA 7687 DOST scholar, and I was also part of two organizations, UP ACME and UP DOST-SA. And right now, I am a volunteer for Queer Scientists PH. In the present, I'm currently working on projects such as in silico toxicity analysis of metabolites from Philippine flora and the research, which I will be talking about in a while. So with my experience on why I took up computational chemistry, so actually at first I was hesitant because of my lack of knowledge in programming languages. But when I took a subject with Sir Ricky, I was encouraged na even though steep yung learning curve, as long as you are really dedicated to learn computational chemistry, you would be able to do computational chemistry experiments and studies. Okay, so let's start. So next slide, sir. Okay, so first, I would like to emphasize briefly that the continuously growing world population, which is actually estimated to be around 9 billion by 2050, means a growing demand for agriculture. So this also means that there is a greater demand on modern agriculture through improved agrochemicals, seeds, fertilizers, etc. And one of the problems that the agriculture industry is facing is the destruction of crops because of pests. Okay, so next slide, sir. Now, one of these agriculture problems that we are facing is the rice weevil infestation. Now, what are rice weevils? So, the rice weevil is scientifically known as Cytophilus oryzae, and it is one of the most destructive pests of cereal products. So, here in the Philippines, they are one of the insects that Filipinos refer to as bukbuk. They attack rice, grains, and other crops by feeding and multiplying in stored grain. So these creatures attack fields in vast numbers, and they have destroyed enormous amounts of crops in the past. So next slide, please. So to mitigate these problems, the usual approach is the use of pesticides to protect crops. But unfortunately, majority of these commercially available pesticides are toxic to the environments and even to us humans. So next slide. One strategy for safe pest management to combat rice weevils is the use of essential oils from plant materials as biopesticide. And they are shown to have activity against certain pests. And now the challenge is what would be the target of these essential oils or these plant materials? Okay, or in short, how to make them effective by starting to understand how they actually function as a pesticide. Okay, so monoterpenes, which is a component of essential oils, have been shown to possess activity against certain insect pests. They are shown to be safe for insecticide because, again, they are from plants. And according to literature, when researchers tried to understand why do monoterpenes possess activity against insects, it was shown that it is because they have some kind of neuromodulatory activity and neurotoxicity because of their ability to interact with the protein targets, such as octopamine receptors. Next slide, please. So one example of these octopamine receptors is the tyramine receptor. So now, if you recall, receptors are protein structures in your body that are responsible for receiving, transducing signals throughout your body system. So in the rice weevil, the function of the tyramine receptor is known to be the invertebrate counterpart of norepinephrine in us humans, invertebrates. So it functions as a neurotransmitter, neuromodulator, and neurohormone. And it is also associated with many important physiological functions such as locomotion, reproduction, and pheromone response. So what else makes this tyramine receptor more interesting? It is that these receptors are abundant in insects, but it is absent in vertebrates. So which means that if we make a pesticide that targets this receptor, it has a low chance that they will also target vertebrates like us humans because we do not have these receptors functioning in our bodies. So this makes the tyramine receptor a desired biopesticide target due to its absence in vertebrates together with its role in insects' physiological and cellular response. So next slide. Okay, so to simplify the concept leading us to the objective of the study, let us use a lock, key, and stick analogy. So let us depict the tyramine receptor as the lock, the tyramine or the activator to activate our receptor as the key to unlock it, and the biopesticide that we are looking for as a stick that fits into the lock but obviously will not unlock the lock. Okay, so next slide. If we use the key, or when tyramine binds to the receptor, then signals continue to be received and transduced, and therefore the insect's functions continue. The insect lives. Next slide. But if we put a stick that also fits into our lock, but it does not unlock it, not only it will block the key, but it will also keep our lock locked, right? So the functions such as locomotion, reproduction will eventually stop, and later on, when the rice weevil cannot do its normal functions anymore, it will eventually die. Okay, so this leads us to the objective of our study, so which is to understand the basis of the monoterpene interaction with insect receptors. Specifically, to understand the basis of interaction, or how the Cytophilus oryzae tyrosine receptor and monoterpenes interact through computationally understanding the pharmacological features of these interactions. Okay? So in order to do this, the following computational methods were used. So first is homology modeling, molecular dynamic simulations, molecular docking, 3D Q-SAR modeling and analysis, and modification of lead compounds and molecular docking, which actually Sir Ricky have already mentioned a while ago, but let us discuss them one by one. Okay, so next slide. So in homology modeling, we use the amino acid sequence and experimental 3D structures of homologous protein to our protein in order to create a 3D structure of our desired protein, which is the rice weevil tyramine receptor. So why is this step needed? It is because the 3D structure of the tyramine receptor of rice weevil is not yet experimentally obtained. That is why we need to model it using the amino acid sequence available in online protein databases and online tools. Okay, so next slide. So after we obtained our model, so we subjected it to molecular dynamic simulations as just like what Jokent did a while ago. So this is because a protein through time changes its conformation, meaning it changes its pose through time. So it is important that we consider that our tyramine receptor doesn't only have one conformation in a real system setting. So after molecular dynamic simulations, we were actually able to obtain 100 protein conformations, or we could see it as 100 poses of our proteins, which we will call now protein ensemble. Okay, next slide. Now, using these protein ensembles, we will now test our monoterpene compounds with them through molecular docking. So, molecular docking is where we test if our monoterpenes will fit or bind into our protein, or as we used in our analogy a while ago, if these monoterpenes will be a suitable stick to fit into our lock. Okay? So after we performed molecular docking, specifically ensemble docking, because again, we have 100 protein structures, we found out that these four monoterpenes right here, terpinolene, carvacrol, carine, and pilgone, are the top hits, or those that exhibited the highest binding affinity to our tyramine receptor. So these top hits were seen to have a binding energy of negative 6.8 kilocalorie per mole. Okay, next slide. So looking at the interactions with the receptor, the top hits were found to share the same binding pocket, or meaning they binded in the same area in the tyramine receptor. So the high binding affinity of the ligands were recorded from the close interaction with these residues. So what are residues? So residues are what makes our protein up. Okay? So these protein residues binding was possible because the interaction with our monoterpenes and residues are what makes the binding possible. Okay, so these protein residues are the most frequent close interacting residues that are common to the four top hits. Meaning sila po yung pinakamadalas na pinakamalapit na nag-interact with our monoterpenes doon sa area kung saan natin siya nakitang nag-bind. Okay? So these results show that a hydrophobic or water-fearing motif meaning hindi siya hydrophobic or water-fearing, is what makes the binding favorable because the interactions were majority hydrophobic fragments. So these residues, these protein residues are mostly hydrophobic. So additionally, the aromatic residue of tyrosine 410 is crucial in the binding of the four top hits. It was also inferred that carvacrol has a distinct hydrogen bonding interaction with aspartate 114, which is a residue. Okay, next slide, please. Now, in 3D QSAR modeling and analysis, our findings from molecular or ensemble docking were confirmed. Now, how? So, 3D QSAR first stands for three-dimensional quantitative structure activity relationships. So, it is where we relate the structure of our monoterpenes to their binding with the receptor and see their correlations or similarities with each other. Now, in this model that we have generated, in the middle are the aligned poses of our four top hits. Or when we say poses, ito yung pose ng compounds natin when they binded to our protein receptors, to our cytophilus erythrae receptor. Okay, so they were aligned. And then here, the green areas in the model shows areas where steric groups should be added or hydrophobic groups should be added. The blue sites are where positively charged groups should be added. And originally, 3D QSAR models would also show areas na red or yellow where we should add negatively charged functional groups or where we should avoid steric groups respectively, but here there is no need for such features. Yun yung sinasabi ng model natin. So the model then suggests that the similarity between the top hits activity or yung binding nila were mostly due to their hydrophobic motifs because as you can see, the model shows a lot of green areas where hydrophobic or steric groups are preferred. And additionally, it also showed that the carboxyl group a while ago in aspartate 114 that interacted with carvacrol acts as a hydrogen bond donor because of the placement of the blue areas. Okay, next slide, please. So, using the findings from ensemble docking and 3D QSAR analysis, the four top hits were functionalized or edited as shown in the figure. So, since both docking and 3D QSAR analysis showed that the interaction is mostly hydrophobic in nature, different hydrophobic groups were added. So, nagdagdag tayo ng hydrophobic groups para tumaas yung pag-bind nila. So, the leftmost structure are the original structures ng four top hits of the four top hits, and the succeeding structures were where we added different hydrophobic groups. So, these structures were docked. So, we performed again ensemble docking to the protein to compare where if there will be any difference when we add these hydrophobic groups to our monoterpenes. And next slide, please. So after we dock and after the edited ligands where we added hydrophobic groups were docked again, the range of the binding affinities changed, where the functionalized ligands were negative 4.7 to negative 8.3. And the first ensemble docking ranged only from negative 4.2 to negative 6.8. So this showed that the lowest binding affinity and the highest binding affinities increased. It is because binding affinities are actually measured wherein the more negative means the stronger or the stronger the binding. Okay, so our binding affinities increased. So after adding the hydrophobic groups. So this, in order to assess statistically if this increase is actually significant and to what degree, the paired t-test was employed. So this is a statistical procedure to determine if this difference in binding affinities is significant and as to what degree. Okay, so next slide, please. So interestingly, so results of the paired t-test showed that the increase of the binding affinities due to functionalization are generally significant. So this confirms that indeed, addition of hydrophobic groups are crucial to the binding to the tyramine receptor as the increase of hydrophobic groups increases binding affinity as well. So in short, pinakita nito na yung increase sa binding affinity is actually significant dahil sa pagdagdag natin ng hydrophobic groups doon sa initial monoterpenes natin. Okay, so next slide please. So among the functionalized ligands, the carine analog, this carine analog encircled right there, is exhibited the highest binding affinity. So siya yung may pinaka-negative, most negative, binding affinity of negative 8.3 kilocalorie per mole. So this carine analog is bound to the same binding pocket or same area as the base molecule. So the improved binding affinity is again seen to be due to hydrophobic functional groups such as okay. So the interaction of the added hydroxyl group to aspartate 114. Okay. So from the binding energy, the inhibition constant of this carine analog was computed, which is actually interestingly surpassed the known activators of octopaminergic receptors. So what we're saying here is that this carine analog was computed na mas mataas yung binding kesa sa mga known na nag-activate kay tyramine receptor. So not only it binds to a receptor, nalagpasan niya pa yung mga known na nag-unlock kay tyramine receptor. Okay, so next slide please. So now in computational chemistry, we can also try to predict the toxicity of a compound. So this table summarizes the results of the toxicity risk assessment done in Osiris Property Explorer. So it is a toxicity software prediction tool, wherein here in the figure, the red cells means toxicity risk is highly present, orange means toxicity risk is moderately present, and green means toxicity risk is not present. So as you can see, the table shows that majority of the top modified ligands are clear of toxicity risks. So which means that they are generally predicted to be safe for humans. And most interestingly, the functionalized carine top hit, which is actually ligand 9675662_5 in this table. Yeah, so right there. So is free of toxicity risks. So wala siyang toxicity risk because there are no red or orange cells there. And additionally, this also supports the non-toxicity of monoterpenes and their analogs in general because majority of the cells are green or free of toxicity risks. Okay, next slide, please. So to wrap this up, okay, in this study, an attempt to understand the key binding features in tyramine monoterpene binding was done using in silico or computational methods. So findings from the ensemble docking show that key residues that are involved are mostly hydrophobic, which was also validated by our 3D QSAR model. Okay, next slide, please. And this research suggests that the carine analog that we were talking about a while ago is a potential inhibitor of tyramine. It was seen that the alkyl hydrophobic groups and hydrogen bond acceptors of carine can improve the binding affinity of our compound to our tyramine receptor. Okay, so this suggested analog is predicted to be free of toxicity risks, and so this carine analog is a potential biopesticide against rice weevils. Okay, so next slide, please. So some current paths for this research are to synthesize this carine analog. First, we predicted, okay, there's a tool online where we can predict its synthetic accessibility, wherein we can predict how easy it is to synthesize our compound that we just edited. And from a score of 1 means very easy, and 10 means very difficult, we got a score of 4.21. Okay? So repeatedly functionalizing the ligand, meaning going over the methods that I have already presented again for another round, is also suggested in order to get a higher binding affinity ligand. So in the present, we are conducting molecular dynamic studies to further understand the interaction of monoterpenes, so not just in rice weevil, but also to other insect pests. So it is important to note that in vitro and in vivo experiments are still required to verify the findings in this work, because as Sir Ricky also mentioned a while ago, this computational approach could only aid in further understanding the mechanisms, and these are predictions using a theoretical approach. Okay? So this computational approach could aid in further understanding the mechanisms and effectiveness of monoterpenes as safe biopesticide for rice weevil and other related pests. And this work is already published in Springer Nature Applied Sciences and is available online. So if you would like to know more about this research, you may visit it online, or you can also scan the QR code on the screen to visit the article. And lastly, we would like to thank the following. So DOST COR facility and UP Diliman CSRC for providing the computing resources. And this project was funded by the Department of Agriculture Biotech Program and UP Vice President for Academic Affairs Emerging Interdisciplinary Research. And that would be the end of my presentation. So if you have questions, you may drop them.
Thank you, Almira. Sige, tapusin ko lang.
Okay, sir.
So, yun yung ginawa ni Almira at saka ni Jokent. Thank you. So, madami rin ginagawa sa lab. Ang daming pwedeng application ng computational chemistry. Depende lang kung sa interest ng students, gaano kadami yung students na gustong gumawa. So, for example, Adam is working on PKR, or this is a drug for schistosomiasis. Okay, gumagawa din siya ng computational protein engineering. Itong sa BKR is in collaboration with Imperial College London under the Newton program. And then, si Kier din tumutulong more on the complexity of the entire machinery of the BKR. So, merong extracellular, intracellular, and the entire ensemble. Si Morris naman, ginagawa niya yung pagdi-design ng compounds or drugs para sa schistosomiasis na to. And not only doing the theoretical part, gumagawa din siya ng experimental using the isothermal titration calorimetry. Ito yung pagtitest kung totoo ba yung magandang nakukuha namin sa computational. And then Kevin, yun yung ka-pair ni Almira in doing this one. But highly theoretical yung kay Kevin, not only looking at the compounds for this rice weevil repellent, but also understanding the entirety of the molecular systems or the molecular machinery. Meron din kaming atmospheric na application. This is done by Roosevelt. He's from TSU, Tarlac State University, at nag-MS din sa UP Diliman. And this is quite complex. Okay, nung nag-PhD ako, hanggang 3-component system lang ako. And for him, he's doing 4 to 5 component system. So this is way beyond. Ito lang yung ginagawa sa buong mundo na sobrang lumampas na ng 3-component system. Si Ronnie, got his MS from Chemen University on QMMM. Ito yung mga reactive na molecular dynamics. Pero ngayon nasa akin siya, and then we will be implementing the Philippine-Taiwan collaborative research on looking for drugs to combat COVID-19. And then si Rashid, sobrang theoretical yung sa kanya, pero maganda itong magiging result nito. Basically, aalamin namin yung using machine learning, aalamin namin yung structure ng mga amino acids sa gas phase at complex solvent systems. And hopefully, if we have a series of amino acid or amino acid sequence, we can predict the structure, 3D structure of that system. Protein engineering naman kay Meg, theoretical, tapos pumunta siya ng Taiwan, in-express niya yung totoong protein na in-engineer niya and anong magagamit noon sa industry. And then si Darwin, ito yung pasimuno ng aming schistosomiasis project, taga Santa Fe, Leyte siya. So gusto niya makahanap ng drug or ng gamot para siya sa schistosomiasis kasi rampant sa kanila yung problem na yun. So we collaborated with the Imperial College London in UK and used the Newton Fund to have this experiment or these studies. And hopefully we can release our results soon with that drug. And then si Peter naman doon sa allosterism, yung complex na drug action. So na-publish na rin niya to. So you can look at his work. And then si Jem naman, tinitingnan niya yung mga superbugs, yung future na mga problemadong sakit na wala tayong mga gamot. And then nagdi-design din siya ng gamot. And then in collaboration with the lab of Dr. Portia Sabido, sinisynthesize din nila to at tinatry kung nag-work. And then, meron din kaming single molecule magnets. So, si Annalie ang gumagawa ng single molecule magnets in collaboration with Academia Sinica in Taiwan and the National NTU in Taiwan also. And then, naglalaro din si Miguel. Si Miguel ay med student na nilalaro niya kung paano i-confuse yung interaction ng human at saka virus for HIV. And then, si Nico naman on lupus, search for lupus cure and understanding lupus. And si Wilvin ay working on understanding Alzheimer's using his systems. So, mga future namin gagawin, yung intrinsically disordered protein, yung protein na akala mo walang structure tapos biglang magkakaroon ng structure. So, example nito yung spider venom proteins na medyo nagagamit for antimicrobial protein or antimicrobial application, at ito yung theme ng mga projects ng UST. So in collaboration with the UST Pharma Group or Biochem Group. So ginagawa din namin. And then yun, yung COVID-19 namin. So hindi namin magagawa to kung wala kaming collaborations. So we have collaborators from inside UP. So we have Dr. Yu, Dr. Junio, Dr. Martinez, iba-ibang fields of expertise sila. And then we have collaboration outside of UP, mga taga-ano naman, SUC. This is in Cavite State University with Dr. Matel, Seya Matel. Si Leonard yung aming focal person dyan. Collaboration with Imperial College London with Dr. Baez. We have collaborators from University of Tokyo, Academia Sinica, Tarlac State, and Cavite State. And hindi namin magagawa to kung walang pera. And this is funded by Philippine government, both national and international. And that's what we have for now. And then, para magtrabaho na maayos yung mga bata, dapat hindi nagugutom. And thank you. We're now ready for questions.
Ayan. Thank you very much, sir. So nakikita naman natin talagang very well and thriving po yung research group ni Sir Ricky. Ayan. So talagang hindi niya kayo pababayaan pag kayo yung ano. So
Kuya Ricky, congratulations po. You built this computational empire.
So amazing.
In the 6 years po. Napakaraming projects po ni Kuya Ricky, so for the benefit po ng mga estudyante at mga guro natin na hindi masyadong pamilyar, for a professor to actually kick off and essentially originate that many number of projects, bihira po siguro sa Pilipinas ang ganyang karami ang nagagawa in just like a short period of time. So, Kuya, congratulations. But JP, before we actually go to the Q&A, I would just like to remind people of a few important dates. So, Kuya, I'm actually just like taking, huwag kayo magulat, I'm taking over the screen sharing. Can you see my JP?
Right, yep.
Okay, so today po, we were able to cover the special topic on computational chemistry. And so napakaganda ng presentation nila Kuya Ricky, ni Jokent, at saka si Almira. Next week, we have the deadline for submission of entries to FilSciHub Research University's Lions Den. So this is the Battle of Science Investigatory Project Idea. So this is specific to high school students. Check out the details on our website and on our Facebook page. So andun po yun. And then also Saturday next week in the Philippines at 10 in the morning, we will have our 7th or 8th formal training course in research, and that is focusing on science communication. So dito po we'll cover best practices in writing up laboratory reports and writing up scientific reports. Kung gusto nyo mag-publish, kapares ng ginagawa ng group nila Kuya Ricky. So this is actually a training course specifically created for both high school and college level science writer. So dito po tuturuan namin kayo ano po yung mga for the past 15 years that in particular that I've been doing this, ano dito ko po ibibigay sa inyo yung mga best practices on how to effectively come up with a compelling scientific write-up. And then on May 29th, we are actually privileged to have Professor Raymond Monterrey, who is a seasoned chemical chemistry educator po. He'll talk about grayscale approach to reasoning in general chemistry. So ito, yung tamang mindset, yung tamang approach kung paano ba talaga maintindihan yung chemistry. And he'll demonstrate this by talking about bonding in solid. So ito siguro pang senior high to, no? Senior high school chemistry topic. Yung mga nasa junior high are also encouraged to attend. And then lalo sa college level, ito malaking tulong to, napakagaling din yan si Sir Raymond Monterrey. And then on June 5th, then the U is coming back and she will be introducing our high school and college students to the world of alkenes. And then also embedded in this lecture is an introduction to reaction mechanisms. Ito po ay aking small participation dito. Tuturo ko sa inyo kung paano ba talaga paikot-ikutin yung mga electrons na yan. And then on June 19th, we will have Professor Katrina Vasquez de Paz of Adamson University. She'll talk about green chemistry, in particular the principles and initiatives of green chemistry. So if you're interested in participating in all of these upcoming web events, feel free to just check out our official web page because nandun po yung sign up sheet. So, all right. So Q&A.
Q&A. Yeah, our audience are very pumped up sa presentation nila Sir Ricky. Ang dami nating questions. But I could probably just start chronologically here from Zoom. So June Alexis Santos has a question here. I don't know if June Alexis is around to...
I asked him. He prefers for us to just read his question.
Okay. So he was asking, is it ideal to perform energy minimization or any structural modification to the protein structure that we want to inhibit right before performing the actual docking process? So I think anyone can answer.
I can answer po yan, sir.
Okay.
Go ahead.
Yung structures po kasi deposited in protein data bank or other sources for protein structure. Most common method for determining protein structure involves X-ray crystallization studies. And in this method po, we cannot determine yung position of your hydrogen atoms in your protein. So, kapag download po natin yung structure, wala siyang hydrogen atoms. And then when we prepare yung protein for simulation, we of course have to add these hydrogen atoms. And then because predictive yung pag-add ng hydrogen atoms, most of the time po actually, nagkakaroon ng bad contacts between these hydrogen atoms, which results po in really bad simulations. So to prevent po these bad contacts, we have to perform minimization studies muna ng ating proteins before yung docking studies.
Okay.
Yeah, so in general, kailangan ng minimization ng energy para makita yung low energy, local energy.
Yeah, and Ricky, siguro dyan papasok yung yung chemical intuition na kuya.
Yes. So dapat merong parang nasa local minimum siya. So dapat minimization. But if you are with a collaborator, you have to also talk to the collaborator. Like for example, pag sinabi ng experimentalist mo na collaborator, gusto kong makita dito sa initial na crystal structure. So basically, crystal structure are arrested structure, different conditions siya. So may solvent effects, lahat na effects nandoon. So yun yung nakikita at that specific time. Pero pag wala kaming alam, walang crystal structure, galing sa homology modeling, the only way to capture that is through statistical mechanics. And statistical mechanics says if you have a lot of structures and then you do experiments on that, maybe your results within uncertainty could represent the entire good stable structures. So yeah.
All right. Siguro, ako naman magapa to JP.
Yep.
So there's a couple of questions from Zoom and YouTube. I actually just compiled them into one document. So I don't know where some of these questions really came from. Siguro question from Patrick Lejan Danan. He was asking, are there in silico methods to determine pharmacokinetic and ADME parameters?
Almira, go ahead.
Okay. Yes po, there are in silico methods po. So, yung ADME po is yung ADME or yung absorption, distribution, metabolism, and extraction po. So yes po, we have available softwares and we also have available in silico tools online website wherein we can predict actually the ADME properties of a compound. So yes po, meron po naman na in silico methods wherein we could also use QSOR actually to also predict the ADME properties of a compound. I hope that answers the question.
Can you tell us the specific software?
Okay. So specific softwares for free na online, we could use Swiss ADME. We can also use pre-ADMET. Okay, so those are online available tools. And for softwares, we can use Tox3. We can also use Osiris Property Explorer, which I also discussed a while ago in my research.
So sa lab, puro kami freeware. Cheap ako. Ayoko nang medyo nang bumibili at medyo magastos yun. So, yeah, freewares lang tayo para at least pag itinuturo natin sa mga estudyante or sa collaborators, walang involved na pera masyado.
Okay. Then, Kuya, this is a question coming from YouTube now, from Renish Simon. His or her question is, what's your advice to people who want to self-study computational chemistry? What are the first few steps to learn computational chemistry? Siguro
okay, sa students na lang. Go ahead, Almira.
So, for my experience po, I mainly studied po using YouTube videos. Madami actually seminars and courses po na available regarding sa fundamentals ng computational chemistry. And then once you grasp na po yung fundamentals or yung theory regarding po dun sa methods, you can then search for tutorials regarding yung software na gamitin mo. And most of the time po, yung softwares natin ay may corresponding documentation po. And step-by-step process naman po yung tutorial for that, for those softwares. Yeah, so mostly on the internet.
Right.
Almira?
To add na lang din po, yes po, ayun, marami pong available na tutorials online. And I think the starting point din po before learning all these kasi ma-overwhelm sila on mostly the tutorials are using codes in the terminals. So I think the first step is actually to familiarize din po with what is a terminal or how we can process data using that. So hindi naman po siya sobrang complicated na topic, but I think that is a good starting point since these tutorials, most of these tutorials, YouTube or online tutorials use na po these types of these codes. So I think yun po yung starting point to be introduced with what is a terminal and how you can incorporate it with your working OS system.
Perspective
naman ng PI.
Yep.
Medyo
and then siguro Kuya Ricky, no? I think I remember you mentioned one very important aspect earlier that their mastery of chemistry should also be good, di ba Kuya?
Yes, it could be. Pero huwag maging stumbling block. So dapat lang, kung hindi naman kaya namang basahin. Nandiyan naman yung mga resources. So ang ginagawa ko sa lab ay independent work. Meaning to say, kung gusto mong matuto, have your best, okay? Search for whatever information you want to learn. So yan yung ginagawa nila. And then try to collaborate, meaning to say, magtanungan kayo among students, among RAs. And ibig sabihin, pag dumating sa akin yung problema, sobrang lala na. So wala namang masyadong dumadating sa akin na problema. So sa akin is pagbato lang ng projects or pagbato ng timeline paano matatapos yung project. At kung may mga concerns na hindi kayang isolve within the perspective ng collaborations. And collaborations involve na rin yung thread sa mga online kasi madami din naman gumagawa ng mga softwares at na-experience din nila yung problems nito. So kaya magtanungan international. So yun lang.
Alright.
Okay. So from IV Joyce Buwan, does molecular dynamics make the protein to be in its most stable conformational structure? Then if you will conduct the molecular docking, how will you direct the ligand that is specific to bind on the protein's active site? I think si Jokent, was it Jokent or Almira,
Okay po.
who was mentioning about protein ensembles earlier?
Yes, I can answer po yung question na yun. So,
Go ahead.
for molecular dynamics po, basically what we do in molecular dynamics is we sample yung conformational space ng proteins natin. And then, once we sample po yung conformational space, may tendency po na masample natin, or mas malakas yung tendency to sample states that have lower energy po compared to other states na may higher energy. And these lower energy states po tend to be local minima or local conformations that are more stable than others po. And then for the next question po, for the docking site, we actually have two different types of docking. One is blind docking and another is yung directed docking. So for blind docking, we do this kapag hindi rin alam kung ano yung site for docking study and mas high throughput yung docking studies using this method. And then we also have the directed docking. We do this kapag alam natin yung active site ng protein, like for example yung protein ko, alam ko yung active site is within dun sa zinc ion. So most of my simulation, lahat ng docking studies ko directed towards that site. And you can direct this using yung mismo docking software. Like you can dictate yung size ng maging docking site.
Dagdag lang ako, in general, when we do MD, supposedly ang gusto nating tingnan ay i-sample yung buong conformational phase space. Pero ideal yon. Okay, wish lang natin yon. Pero napaka-rugged ng phase space ng proteins. Therefore, meron siyang energy barriers. At kung normal molecular dynamics lang yung ginagawa mo, pwede ka ma-stuck doon sa local minima. Hindi ka maka-jump doon sa barrier na yun. Therefore, you cannot sample outside of that paradigm. So meron din namang mga enhanced MD na mga techniques. Pero sa amin lang, normal MD lang. So in short, yes, makakakuha ka ng local minima. Pero kung gusto mong ma-sample yung buong phase space, ideal case.
Alright.
Siguro ito isang question. Ito Kuya Ricky and everyone. No? This is coming from, I think, a high school teacher. His or her question was, siguro nanghihingi ito Sir Kuya ng broad ideas. No? Anong simple research topic areas under computational chemistries ang pwedeng gawin ng mga nasa high school? So they were also asking siguro yung mga software na accessible. So at the high school level, if they would like to venture into a science investigatory project, meron ba ko yung mga topic areas na pwede nilang i-explore na pasok sa high school level?
Meron naman yung level nung kay Almira, pero wala lang molecular dynamics. So basically, pwede kang merong different insects or whatever available na mga crystal structures sa PDB. So kuhanin mo lang yun and the different compounds, docking ka lang. Either directed or blind docking. So kaya naman yun. And we can collaborate, we can assist on that.
Grabe Kuya, I just wanna highlight the fact that when we were in our undergrad years. Siguro this was about siguro 16, 17 years ago. Ang tanda ko na yung docking thesis na yun sa sa UPLB. So it's just quite amazing how fast, like the area of computational chemistry is really advancing. So such that pwede na siyang magwata high school level. And yun kuya, sabi niyo you guys can collaborate on that. Ito follow-up question ko lang. This is my own question. Kuya, do you also conduct training courses or webinars for specific for high schools?
Yun yung plan namin with the group. Baka sa mag-summer workshop tayo just for high school. We'll start it with science and technology na group, STEM ata. And then, focus lang muna tayo sa science-oriented na mga schools and then palawigin natin papuntang normal.
We can promote that anytime.
Sure. Sinusort out lang namin kasi medyo in terms of software, in terms of computers, kailangan yung requirement.
All right.
So, meron kaming tawag dito, donation from Taiwan. Taiwan gave us around 2 million. So, merong computer laboratory ang UP na pwede sanang gamitin. Kaso wala tayong face-to-face ngayon. So, baka pagdating ng panahon, gamitin natin yun. And I think Ched would be interested to help on this kind of endeavor. Mm-hmm. Yeah, so tingnan natin in the near future. But immediate, I think, magkakaroon by maybe June, July.
All right.
Initially, pinlan namin ng May, pero hindi kaya. Medyo sobrang daming ginagawa.
Yeah, all right. Ako sa ating mga live participants po ngayon dito, watch out for announcements. So may magandang plano po ang Good Vibes Lab. So definitely napaka-good vibes talaga, no? Napaka-good vibes yung mga sinasabi.
Napaka-good vibes talaga.
Yeah, congratulations and thank you so much for your planned activities for our high school sector. So JP, do you have other questions?
I have a number of questions here from Miss Melanie Ann Munoz. I think she's around to ask her question live. Ma'am, can you unmute and... Miss Melanie?
Good afternoon po or good morning. So for my first question po, dun sa first presentation, for the reported average binding energy values, is it computed within the number of binding poses po? Or within all the trials or replicates?
Almira or Jokent?
Is it yung question po for my presentation?
In general, you can answer that.
In general. Okay. So yung average binding energy po was computed because, again, just like what I have said a while ago, yung protein po natin are ensemble. So we have 100 of them. So we computed for each monoterpene yung average nila na binding energy for those 100 protein conformations. So meaning one monoterpene, tinest po natin siya kung kakabit po siya doon sa 100 protein conformations, and then each conformation, each parang pair ng monoterpene and protein conformation has a binding energy. So we computed the average ng binding energies na yon, and yun po yung nire-report namin usually instead of reporting yung binding energy ng each of those 100 conformation. Ayun po. I hope that answers the question.
It answers naman. Wait.
Yeah, I think you still have a few others there.
So for the second question po, kasi di ba po, there are a lot of available programs and web servers online. So is it wise in your study to use more than one to validate your results?
Mm-hmm.
Pwede naman po. Pwede niyong gawin yun as a self-experiment para ma-guide kayo or ma-convince kayo na tama nga, kahit anong software ay medyo pareho sila ng nire-report. But you have to understand, bawat software, meron silang sinasabing uncertainty ng measurements nila. So within that error, kung makikita mo bawat software within that error, ay medyo magpareho lang sila. Because I think depende lang kung paano nila tinitreat yung interaction ng ligand as well as the receptor. So I would suggest, yeah, you can do that for parang na rin, experiment na rin with your students or with yourself. Pero kung production level na, there are a lot of publications and there are softwares na medyo ginagamit na usually. Pag pinublish mo siya, under review siya, sasabihin na ng mga reviewer na no question on this kasi tested na siya before. But yes, you can do that if you want. And yung number 3, sagutin ko na, other than the number of possible protein conformations, what other information can be generated from MD simulations? Depende po sa inyo kung anong gusto nyong tingnan. Pwede ka ring makakita, for example, tingnan mo yung hydrogen bonding. Merong mga hydrogen bonding na nag-on-off. Baka meron pala yung gamit doon sa dynamics ng system. At saka, tingnan mo kung meron ba talagang hindi lang conformations, kung hindi, parang pwede mo na siyang i-deal na medyo reactive system siya or nagbabago talaga yung oxidation state. Hindi namin masyadong ginagawa tong mga ganitong uri ng MD projects, pero I think yung force field ni Jokent yun yung tinitingnan. Copper 2+ naging copper or something like that. Meron ding mga reactions, halimbawa, pag dumikit yung isang drug doon sa receptor, pwedeng may catalytic reaction, nagbabago yung anyo ng ligand. So meron ding mga ganun po. At medyo pang PhD na po yung level na yon or pang industrial application. Mahirap pong ganun ang training natin sa mga estudyante kasi baka biglang mawalan sila ng pag-asa, hindi na mag-proceed. So yan, ingatan din po natin yung ganun. Challenging experiments or questions, that's okay. Pero to the point na medyo matcha-challenge ng sobra or mag-breaking point na ng mga estudyante, medyo iniwasan ko rin yung gano'n.
Siguro, at Kuya, sisingitan ko lang ito isang question naman from Ivy or Contigua. Conchuga, I don't know how to read her name. Essentially, are there possibilities of applying computational chemistry in geochemical research?
Yung isa kong collaborator, I think yun ang ginagawa nila. So, yung mga geomaterials natin, formation of those materials, medyo may chemistry din yun. Ang problema lang nito, this will happen in a time scale na sobrang mabagal. At saka, in a condition where high pressure, high temperature, maybe extreme condition. But yes, it can be done. And there are people who are doing that. In fact, one of my co-PhDs sa group namin, I think nasa Corning na siya. I think yun yung ginagawa nila. Mga silicates na computation.
Siguro, ganyan.
So industry at saka geo-monitoring. Kaya naman, pero hindi namin ginagawa. Sorry. Not within our expertise.
And then, siguro ko, ito isa pang question, no? Siguro this person is asking about yung limitations, siguro, Kuya, or margin of error when it comes to computational chemistry. So his question was, do you conduct wet chemistry analysis? Siguro ito, Kuya, mga experimental confirmation. And are there any instances that there is or are deviations in the results?
Yun ang una kong sinasabi, na when you do computational chemistry, in real life, we only get insights. If that's the reality, it could be. But let's be careful about that. It could be insights. And that's why we always collaborate with experimentalists, MSU, IIT experimentalists from UP Diliman. And when we do this, we have results from theory or computation, and we have results from experiments. And after having all those results, we do correlation analysis. Right. And correlation analysis could have a weak correlation, mid correlation, or strong correlation. Happy ka na kung meron kang strong correlation, but expect a weak to mid correlation.
And actually, highlight pwede namin din i-highlight. Karamihan din po ng mga research namin ngayon abroad are also aided by computational chemistry. So, yun po, maganda po talaga may balance yung approach. So, gaya na sinabi ni Kuya Ricky, mas maganda kung kapag meron kang experimentalists that you collaborate with. So, typically po, mabagal tayo sa experimentation kasi pa isa-isa lang tayo, pero they can accelerate our progress sa screening ng possible outcomes or sets of conditions, no? Pero yung computational aspect naman po, you also need the insights of experimentalists to actually make sure that the output that's being generated is really something that is what they call this, reproducible then on an experimental basis. So
yeah, in connection with that, Kuya Jeff, may tanong si Peter James Dakinya. I think this is directed to the Good Vibes Lab. If they conduct wet chemistry analysis, and if there are instances wherein there is the deviation between the computational data and the experimental.
Yung ginagawa lang sa lab na wet analysis is the ITC, isothermal titration calorimetry. Other than that, may collaborators kaming gumagawa, synthesis as well as testing or assays. Deviation, yes, there's always a deviation. You have to understand na pag computational chemistry, clean system siya. So wala yung impurities, wala, kaya na may control yung system. Pero pagdating sa experiments, ang daming factors na involved. And that's where the correlation would come in. So, pag highly correlated, ibig sabihin ko anong ginagawa namin sa computational, baka yun din yun ang nandoon sa system. Or pwede ring yung true system hindi na mimic ng computation. So, nagkakaroon ka ng deviations doon sa area. That's why you have to have your chemistry, basic chemistry on hand para kung meron ka nakitang results, hindi ka sasabihin, ay, mali ito or mali yan. But you can infer or you can have some sort of a rationale. Bakit nangyayari yung ganun? Uri ng hindi pagkakasundo ng data. Right?
Clear, Ricky, one more follow-up question kasi siguro ito patapos na tayo sa Q&A. Ang dami yung projects and obviously ang daming publications na lalabas out of these projects. Kuya, do you upload, like copies of these papers on your group website para siguro kung yung may follow-up ang ating mga teachers, may interest area sila, baka pwede nilang mabasa, Kuya, yung output of your projects in the form of scientific publications. Pwede man nilang puntahan yun, Kuya, if you have an official group website?
Pwede naman yun, pero hindi kami naglalagay ng sort of bank ng mga publications na to kasi alam natin yung meron kasing mga issues sa ganun. So may mga libre din naman yung binabayaran namin.
Open access, Kuya.
May open access yung binabayaran namin na article publication charge. So open yun. And if you want one, pwedeng directly you can email us.
Ayun.
So email us, we can always send you a copy. Yun ang allowed. Personal sharing.
And I think Sir Ricky, meron silang Facebook page.
You have a Facebook page,
The Good Vibes Lab Facebook page. So pag minsan, I think nakita ko may mga projects silang nakapost doon. So I think everybody can just check it out. Yep. At kung meron po kayong ma-interesan doon sa mga topics nila, then you can just directly contact them. Yep. So I think we can wrap up the discussion at this point. So ginather na lang po namin yung lahat ng questions and we will send them to the speakers and probably we can post like a follow-up for the questions. Ayan po. So at this point, I would like to present a certificate of appreciation. Nakikita mo ba, Kuya Jeff?
Yeah.
Okay. So we would like to present this certificate of appreciation to Professor Ricky Nellas of the Good Vibes Virtual Biochemical Explorations Lab, Institute of Chemistry, University of the Philippines, Diliman, for delivering the webinar Computational Chemistry and its Applications, organized by the Filipino Science Hub and held by Zoom and YouTube Live on the 8th day of May 2021, signed by Dr. Jeffrey Bungkin, CEO and founder, Filipino Science Hub, and yours truly, JP Onya, Head of Research, Filipino Science Hub. And we will also like to present this appreciation certificate to Mr. Joe Ken Gaza, also from the Good Vibes Lab, and as well as Miss Almira Ocampo for being such great troopers today. Maraming salamat, guys. we are now it's 12 noon in the Philippines, no?
Yep.
So I think
Yeah, actually there
yeah.
are like 300 participants over the past, a full 2 hours. So tumutok po talaga sila. I bet ang dami nilang natutunan. Camera on para makapag-picture tayo.
Yep.
Para remembrance lang, like, with our class. Ayun. And then, Marty, could you take the picture, please?
Ayan, pwede nyo na pong buksan ng mga cameras for a photo op.
Alright.
Kuya JP, yung link sa
Yeah, I will post it now.
Sige. Smile po. 1, 2, 3. Isa pa po. 1, 2, 3. 1, 2, 3. Okay na po. Thank you po.
Ayun. Kuya Ricky, maraming salamat po, Almira and Jokent.
Salamat din.
Ayan.
And Kuya, congratulations. This is the first time that I have seen like the magnitude of your work and what you've built over the past 6 years. Like super impressive.
Ayun, kalbo na ako.
Yeah. No, but it's all worth it. And dami po. Ang galing. Sobrang galing. Pati mga estudyante niyo, ang gagaling. Congratulations po.
Salamat din sa funding agency. We cannot do this without the funding agency.
Yeah.
It's the Philippine government's I think DOST and DA Biotech.
All right.
And UP, of course. Yeah.
Alright. Naku kuya, forewarning, baka dumugin na kayo ng mga high school at college teachers and students natin for collaboration. So that usually happens whenever we conduct web events like this. Dinudumog yung mga speakers. So markado na kayo, kahit kayo yung kakapitan nila.
Brace yourselves.
Ayun, kuya, maraming salamat po uli sa share ng oras. I'm hoping this is not the last. Hopefully sometime in the future maimbatahan namin uli kayo pabalik, kuya. Hopefully for another follow-up.
For sure. Sige. Salamat.
And thank you din po kay Jokent and Almira for sharing your research to us this morning. Yes.
Thank you din po.
Very well done. Very well presented. And ayun po, na-post ko na po yung link sa certificates on the website. So kindly check it out. And dun po sa mga registrants natin, so kindly note na lang po na yung registration po is not a guarantee for certificate. You have to fill out the evaluation form na pinost po natin ngayon para makuha po yung certificates nyo. And please wait for 7 days para po ma-deliver namin lahat kasi because we're delivering in batches the certificate. So konting patience lang po sa paghihintay ng inyong mga certificates. Okay? Okay, so at this point I think we can wrap up now. Maraming salamat po again to our speakers, to our participants in here in Zoom and also in YouTube. Thank you for taking the time for joining us today. So maraming salamat po, and next week we will have another course at the FilSciHub Research University about scientific communication. So makikita nyo naman po ang mga pagmumukha namin next week.
Okay.
I think we're gonna start 9 a.m. Ayun po. So
Ayun.
yung mga details po nasa website lang namin at saka sa Facebook. Alright?
Alright. Maraming salamat po ulit. Ingat po kayo lahat. Ricky, thank you. Almira and Jokent.
Thank you. See you around.
Maraming salamat po.
Okay. Have a great
Have a great weekend.
Take care.